Imagine this. Imagine you arrive at the hospital after suffering from a fever and severe headache. Imagine that as you're checking in, someone from the hospital staff is already digging your grave. Imagine discovering that your best case scenario is a 50-50 shot at survival. Imagine being told that you may have already infected your family and loved ones.
Imagine contracting Ebola hemorrhagic fever. For years, this frightening disease has been on the radar of the global health community. We know it's bad news, but what exactly is Ebola? What should we know about the pathogen, and why should we be concerned? There are 4 variants of the Ebola virus: Zaire, Sudan, Côte d'Ivoire, and Reston. Ebola is part of the Filoviridae family, which is characterized by thread-like filamentous structures. The virus usually appears in this tube shape, but can also be seen in a shorter U-shaped, mace-shaped, and ring configuration. This tiny pathogen is only 80 nanometers in diameter on average. The genetic information of Ebola virus is stored as negative-sense single-stranded RNA, which codes for 7 genes. The incubation period can be anywhere from 2 to 21 days. Medical researchers have identified the NPC1 protein as crucial for the Ebola virus to gain entry into the host cell. Once the virus enters the cell and begins replicating, it damages the endothelial cells that make up the lining of the blood vessels in the body. As the vessel walls become more damaged and the platelets become unable to coagulate, the individual undergoes hypovolemic shock, a dramatic decrease in blood pressure. The initial symptoms of Ebola include fever, severe headache, joint and muscle aches, chills, and weakness. Later symptoms of the disease can include nausea and vomiting, diarrhea which may be bloody, red eyes, raised rash, chest pain and a cough, stomach pain, severe weight loss, and bleeding from the nose, mouth, rectum, eyes, and ears. As the disease progresses, it can lead to multiple organ failure, severe bleeding that can be fatal, jaundice, delirium, seizures, coma, and shock. For those who do survive, recovery is still very slow. Some of the long-term symptoms can include hair loss, sensory changes, liver inflammation, weakness, chronic fatigue, chronic headaches, eye inflammation, and testicular inflammation in men. The Ebola virus is initially contracted through contact with primates such as chimps, baboons, and gorillas. From there, it spreads through the human population.
Fruit bats of the Pteropodidae family are considered to be the natural reservoir of the Ebola virus.
Ebola is spread through contact with bodily fluids, including blood, saliva, stool, breast milk, and tears. It may also be spread through other secretions that have yet to be tested. There are currently 2 blood tests used to test for Ebola. The first is enzyme-linked immunosuppressant assay, ELSA. The second is reverse transcriptase polymerase chain reaction, PCR. These can detect specific genes, the virus, or antibodies to the virus in the bloodstream.
Currently, there is no effective antiviral treatment for Ebola. As a result, treatment is limited to supportive hospital care, including fluid IVs, maintaining adequate blood pressure, blood transfusions, and treating secondary infections.
1976 marks the very first appearance of Ebola. The first outbreak was in Sudan, and it was fatal for 53% of the 284 people infected. The second outbreak that year ravaged Yamboko, Zaire, now the Democratic Republic of Congo. 318 people were infected, and the fatality rate was a startling 88%. The third strain, Ebola Reston, wasn't identified until 1989. Monkeys infected with the virus were imported to Reston, Virginia from the Philippines. Fortunately, however, Ebola Reston does not cause disease in humans. Ebola Côte d'Ivoire was discovered in 1994 when an ethnologist accidentally infected herself while performing a necropsy on a dead chimpanzee. One thing that is concerning about Ebola outbreaks is that they are frequently misclassified initially as outbreaks of other diseases. The concern is that misinterpretations of symptoms can lead to the spread of Ebola in medical centers because the necessary precautions are not taken.
As of May 2012, there have been 21 different major outbreaks of Ebola according to the World Health Organization. 1976, Democratic Republic of Congo, 318 infected, 88% dead. 1995, Democratic Republic of Congo, 315 infected, 81% dead. 2007, Democratic Republic of Congo, 264 infected, 71% died. 1976, Sudan, 284 infected, 53% dead. 2003, Congo, 143 infected, 90% dead.
Groups like the World Health Organization put primary efforts in educating about prevention and hospital procedures. At this point, prevention and controlling spread are still our best weapons against hemorrhagic fevers. The tragedy of hemorrhagic fever is very serious and significant in Africa. There, Ebola is feared for causing sporadic outbreaks, spreading easily in medical facilities and to those administering care, and for the fatality rate that reaches up to 90%. The extremely high fatality rate, coupled with the fact that no vaccine or treatment exists for Ebola, provokes the attention of countries where the virus doesn't even exist. These characteristics qualify Ebola hemorrhagic fever as CDC Class A bioterrorism agent. Relatively speaking, incidence of hemorrhagic fever is fairly low, but the global health communities do not underestimate the disease's potential. The importance and significance of Ebola virus goes beyond our concern for the disease. Ebola is also important because of the questions it has raised in the medical communities. Its sudden appearance within the past 50 years makes Ebola hemorrhagic fever a new modern disease. Where did it come from? What made it appear? Should we expect to see more sudden emergences of other new diseases in years to come? The knowledge we stand to gain from Ebola, and all that we stand to lose from it, makes the Ebola virus one of the most important pathogens to research of our time.